Hi my friends, good evening to all of you. So as the exam is approaching up in few days, so obviously I will not be wasting your time in any sort of things. It will be a very high yield session, and from this particular one, if you do it properly and if you are there with me for 1 to 1.5 hours, whatever time we take up for this, so surely from my side 30 to 40 questions you will influence up with this particular one. Okay, so let's start up without wasting time. Hi Dr. Anil. Hi Dr. Dr. Ashwani is also there. Hi sir. Hi Madhav. So let's start up guys with the first image, and as you can see this particular one, a given microbiological instrument is there used for the isolation of. So as you can see this particular picture, and if I zoom it up for you, it is seeing up written up gas pack. Okay, and gas pack, you know guys, this is a chemical method of anaerobiosis, and this chemical method of anaerobiosis is used to create what? Anaerobic environment. And anaerobic environment is used to grow up which particular organisms? That is the anaerobes. Okay, and which of the following is anaerobe here? Clostridium. So everybody knows it. The Clostridium is the anaerobe, and it grows up with the help of this gas pack. And gas pack actually is like a ORS sachet. When you cut it open and keep it into a jar, it will absorb up all the oxygen and it will create what? Anaerobic environment. Okay. So try to tell yourself also the answers and all, and I will be telling you in and around that particular thing, a lot of things. So obviously it will be a very high yield stuff. In the Clostridium, guys, can you tell me up which is the culture media for them? Clostridium, anaerobes. What is the culture media? The culture media for anaerobes is what? Yeah, Dr. Anil, absorb oxygen, very right. Which is the culture media I want to ask for the Clostridium? Yes, anybody in the chat box? What is the culture media for this Clostridium? In which particular culture media you would love to grow this? Yes? Answer is RCM. And what is RCM? Robertson cooked meat media. What is RCM? RCM is Robertson cooked meat media. And this particular meat pieces, there was a question in the Fmge exam, and this particular session is equally good for the NEET PG also. Okay. So if it is there, medicos, it is not a Loeffler serum. It is RCM, Robertson cooked meat media, and these particular meat pieces, okay, these meat pieces, if they turn pink, then that is termed as saccharolytic. Okay, and if the meat pieces turn black, that is termed as proteolytic. So that you must be knowing only. Okay. So Manisha, I've told up right, RCM. So meat pieces when turn pink, that is termed as, I am writing up here, saccharolytic. Okay. What is the meaning of saccharolytic? Which like use of the saccharides, that is termed as saccharolytic. Which use the proteins and all, that is termed as proteolytic. So saccharolytic is Clostridium perfringens. If you take up an example, Clostridium perfringens is a saccharolytic one. And proteolytic one is Clostridium tetani. Okay, so that is Clostridium tetani. Now this question already have come in the Fmge exam, and in fact in one of the NEET PG also this have come, that which turns which color. If it turns pink, saccharolytic, and black, proteolytic. How to remember that? Pink sari, black pants. Okay, black pants, pink sari. So you will able to have some particular resemblance here regarding the meat pieces color change. An example I already told up. Fine. Yeah, very right. Very right. Okay. So this is what is can be asked up in the exam again regarding the anaerobes. Okay. And answer to this particular question is Clostridium. Is it fine? Yeah. Clostridium tetani is slight proteolytic. You are right, Abhinav. But it does change it black. Okay. Now coming to this particular one, a 60-year-old man was brought in with a history of high-grade fever, okay, chills and rigors for the previous 2 days. He had a mild chest pain productive cough is also there. The sputum was submitted for investigation. The X-ray chest shows consolidation in the right lower lobe, a characteristic finding of a particular organism. The direct smear from the sputum showing this picture is being given up down. Okay, on the basis of gram staining, what is the probable cause of the infection? And I like to show you by zooming up this, that you can see up in this particular question, certain things are there which are in pairs, okay, which are in pairs. And what is in the pairs, guys? Pairs is diplococci, okay. What is there in the pair? That is the diplococci stuff, and means they are in pairs. And what is the shape of them? They look like flame shape, also termed as lanceolate. So what we are talking about, guys, what should be the answer here? The answer is Streptococcus pneumoniae. As everybody is telling, right? Answer is Streptococcus pneumoniae. And this picture, already a NEET PG question again can come. They can show you this particular one and this particular arrangement, diplococci arrangement, and this shape, also termed as lanceolate. Another thing which you need to know here in this particular question is this mnemonic. This mnemonic, I think, can definitely be asked, and that is bio. Okay, and what is there in the bio? Bio is, it is bile soluble. Okay, we are talking about what? We are talking about pneumococcus. Pneumococcus is P. P for P. It is positive, bio positive, bile soluble, inulin fermenter. Okay, this particular polysaccharide is being fermented by this, and optochin sensitive. And optochin is a drug, so it is optochin sensitive. Okay. So bile soluble, inulin fermenter, and optochin sensitive. This is what is termed as bio, and bio is positive in pneumococcus, negative in viridans. It was asked in NEET PG. Okay, so bio, these particular biochemical reactions, bio, as the mnemonic tells, this particular thing is negative in, it is negative in the viridans, and it is positive in what? It is positive in the pneumococcus. Okay, remember that. Now, another important thing, the most common cause of CAP. What is CAP? Community acquired pneumonia is pneumo, guys. Okay, is pneumococcus. The most common cause of typical pneumonia. Typical means lobar pneumonia. Again, answer is what? Pneumococcus. Okay, the most common cause of pneumonia in HIV positive. In HIV positive, again your answer is what? Pneumococcus. Okay, so all of them, answer is what? Pneumococcus. And the question which have recently came, that the most common cause of atypical pneumonia, the answer is Mycoplasma pneumoniae. Okay, and that pneumonia is termed as what? Walking pneumonia. And that is the atypical pneumonia, not the typical one. So in HIV positive also, overall most common is pneumococcus only, but when the CD4 count turns out to be less than 200, then comes pneumocystis cerevisiae. Okay, pneumocystis cerevisiae pneumonia at a CD4 count of less than 200, for which you use up which particular drug, guys? Cotrimoxazole. Okay, so very right, everybody is telling wonderfully. Okay, so these are very high yield stuff regarding the pneumococcus which ought to come in your exam. Another one, if I ask you that there is something about the capsule of this organism, what is that particular something? That something is Q-lung reaction. That something is what? Q-lung reaction, and this is basically the capsular swelling. Q-lung, lung, pneumo. So Q-lung reaction is associated with this particular pneumococcus only. So these are the important ones which you need to know regarding the pneumo. Now, again, another question which they can give you, and they can give the features of pneumonia in the question. Okay, and they can ask up the typical pneumonia features only, and they can show you this particular culture plate in the exam. Okay, can you tell me, guys, by seeing up this culture plate only, can you tell that what we are talking about? Yes, anybody in the chat box? What we are talking about, anybody can tell that this particular culture plate is showing up something, mucoidness. I am giving you a hint. There is a mucoidness in this, and mucoidness is a feature of which particular bacteria, guys? Mucoidness is a feature of which particular bacteria? Abdul is on the right track. Okay, Abdul in the chat box is on the right track. It is towards the Klebsiella, guys. So they can very well relate it with this particular picture, and they can show you this McConkey agar. This is McConkey agar, And on this Mekongki agar, you can find mucoid colonies. Okay, and in the chat box, somebody is telling a lactose fermenter. Yes, it's a lactose fermenter, and Klebsiella is going on it. And this mucoidness is typical of Klebsiella. Okay, so as they have given the gram stain picture of pneumococcus, here what they can do up, they can give this and they can ask up that alcoholic is there and it is having the sort of consolidation and all. Okay, and a lobe involvement is present. Which particular bacteria? To Klebsiella pneumoniae. And along with that, the favorite of examiner are 2 points. Everybody knows them. I suppose you must be knowing regarding Klebsiella pneumoniae. Somioli is not non-lactose fermenter. This is a lactose fermenter. This is Klebsiella pneumoniae is a lactose fermenter, not the non-lactose fermenter. Okay. And 2 particular points which I am telling up is what? They are red currant jelly sputum and bulging fissure sign, which is seen on the chest X-ray. They are so favorite examiner. The examiner must have had some time Klebsiella pneumoniae. It have become favorite of them. 20 or 25 times repeated question this is. Red currant jelly sputum and bulging fissure sign is a feature of. It is a feature of Klebsiella pneumoniae. Is it fine? So they can easily relate up with this and they can ask you this particular question. Now you can say, the E. coli can also show these types of pictures. No, mucoidness will not be there in E. coli colonies. Lactose fermenter it is, but mucoidness will not be there. Mucoidness is a feature of Klebsiella pneumoniae. Okay, and if we are talking about Klebsiella, guys, remember these particular things. Regarding the Klebsiella, important thing is Klebsiella, illa, okay, it is non-motile. Illa it is there, so it is having non-motility. It is the important feature of the Shigella, Klebsiella. They are what? They are illa. They are non-motile. Fine. So remember that particular thing also. Yeah, Arvind, very right. Red currant jelly sputum, bulging fissure sign. Utteri, Dr. Anil, you have told everything regarding it. Wow. So gram negative, non-motile, encapsulated, lactose fermenting, facultative anaerobe, rod-shaped bacteria. Okay, you can add one thing more in that if you have written so much. You can add upon it is urease positive. Okay, it is urease positive and it is the one which produces mucoid colonies. One of the common nosocomial infection, this particular fellow is there in that. Fine? No, it's not swarming growth, Chandni. You are wrong in that. Fine. So this answer is Klebsiella pneumoniae, and another one which are important, let me tell you, Klebsiella uzini leads to uzina, that is atrophic rhinitis, rheumy nose and immense foul smell. Anosmia is present in that particular Klebsiella uzini infections, and one is Klebsiella rhinoscleromatis, which leads to rhinoscleroma, that is termed as woody nose. And when you go for the biopsy of it, you find what? You find Miculous cells and Russell bodies. Favorite question again of the examiner. Okay. Normally harmless. Normally harmless. Yeah, you can say they are commensals in some bodies, yes, but you can't say normally harmless it is. Now, coming to this particular one, guys. Identify the hemolysis and the bacteria producing it. As you can see this particular picture, if this comes in your exam, can you tell me what we are looking up to? Merciful. Yeah, very right, Manisha, very right. Now coming to this one, yes, tell me what you are observing in this. Aren't you observing a double zone of hemolysis in this? So this is termed as double zone or target hemolysis. But the important thing is target hemolysis, double zone hemolysis of what? What is the options telling up? Yes. No, Janni, it doesn't show swarming growth. No, it is having a mucoid one. Okay. So tell me this particular one. Double zone of what, Preeti? Double zone of what? Is it double zone hemolysis by Clostridium tetani? Is it target hemolysis of Fmge? Is it beta hemolysis of Botulinum and incomplete hemolysis by Clostridium difficile? Yes, tell me fast. Double zone is fine. Answer is what? A, B, C, D. Yes, what is the answer? Preeti have told A and answer is wrong. Okay. Clostridium tetani does not show double zone hemolysis. Which particular organism does show it? The answer is B, guys. Answer is B. Target hemolysis it is, and shown by which fellow? Clostridium perfringens, which is also termed as Welchii. Okay. So answer to this question is B rather than A. Like this, the examiner will play the tricks, because double zone is also termed as what? Target hemolysis. And another important names of the reactions of this is already we know in our lectures, senior NTR. This is the mnemonic for it. The reactions of Clostridium perfringens, Vulci. S is stormy fermentation of litmus milk. N is Nagler's reaction. T is target hemolysis. R is reverse camp test. Reverse camp test. Okay. So answer to this question is B. Answer to this question is B, and let me revise up very fast the Clostridium. Clostridium perfringens, what does it do, guys? It does gas gangrene. Okay, gas gangrene, Clostridium perfringens, and it is a what? Which type of bacteria? What is the shape of it? Brick shape. Okay, brick shape. If the boat shape comes, then that is septicum. Okay. Other than that, it causes food poisoning. An important point regarding food poisoning is it is the only one which produces heat labile antirotoxin. Otherwise, all others, they mostly produce heat stable. Okay, that is important point. And there is one particular thing termed as pig bell. And pig bell, it is, as the name suggests, eating up uncooked pork. That leads to this. And beta toxin type C does that. Okay. Clostridium tetani. Clostridium tetani, the important question which can come regarding that is this particular one, the tetanospasmin and tetanolysin, which is important, spasmin. And spasmin acts where? Pre-synaptic inhibition of glycine and GABA. Okay, that leads to the constant stimulation. And can you tell me which particular snare protein is involved in this? The snare protein involved is synaptobravin. Okay, synaptobravin. Clostridium botulinum. Clostridium botulinum is the one which have an action of anticholinergic, decrease in acetylcholine at various junctions, like neuromuscular junctions, like postganglionic parasympathetic. Okay, so number of other junctions also it has having action, and the main action is what? Decreasing up the acetylcholine levels. Okay, it leads to canned food poisoning, that you know, antirotoxin is responsible for that. Most of them are neurotoxins, and this particular one, important regarding this is that it is being used for the Botox treatment and all, and most potent toxin on earth that is being told up is Clostridium botulinum. And snare proteins, synaptobravin, syntaxin, and SNAP25. These names, they are coming in your exam. SNAP25, syntaxin, and synaptobravin, these are the snare proteins involved up for the Clostridium botulinum. Okay, telling up regarding flaccid paralysis, Abdul, very good. Okay, telling up Clostridium difficile, also termed as Clostridioides difficile. Fine. CDAD, associated diarrhea, mostly run by which fellows? Third generation cephalosporins, then Clinda, then ampicillin. Which is the drug of choice now for this? That is pseudomembranous colitis and all. Vithoxamicin rather than the vancomycin. Fine. And what is the best way to diagnose this particular stuff? The best way to diagnose this is toxin demonstration by ELISA, along with that GDH. And what is GDH? Glutamate dehydrogenase estimation. So we have discussed up the full Clostridium in 5 minutes. Fine. So I suppose this particular thing will help. These all are questions and they will be coming up in the exam. Fine. Now coming to this. See this particular picture and tell me, obviously it's written up there itself that which test I am talking about, ALEC test. So my question for you is that this particular test is done for which bacteria? First of all, and out of these particular ones, can you tell me which should be the answer? That obvious hai, this is not the answer, this is not the answer, this is not the swarming of it. So obviously answer is this. This is a agar diffusion in 2 dimension. So guys, this is Ehrlich gel precipitation test, which is not done nowadays, but definitely can be asked. Okay, and this particular thing is which sort of a diffusion? Double diffusion in 2 dimensions. What it is termed as? Double diffusion in 2 dimensions. And double diffusion in 2 dimensions, typical example if they ask is Ehrlich gel precipitation test. Ehrlich gel precipitation test, which is done for what? Corynebacterium diphtheria. Okay, for the diphtheria diagnosis, you do this particular one. And if somebody asks you which type of diffusion? Double diffusion in 2 dimensions. Means in 2 dimensions only you can measure. 3D you can't measure. So that's why you say it. And what is it? Toxin antitoxin action. It is toxin antitoxin action. It's precipitation lines they produced up. As you can see up in this, these lines they are there. So non-toxigenic strain you take in that, positive control. Then the patient isolate you take, and a non-toxigenic Corynebacterium diphtheria you take. Okay, and what you observe is when this particular filter paper strip with the Corynebacterium diphtheria antitoxin you take and you produce up these 3 lines, positive control, negative control, and the patient line, you observe the formation of these precipitin line. Okay, So as the percipient line are produced in patient isolates also like this, that suggests, yes, patient is having an attack of toxigenic stuff. It is producing up toxins. Is it fine, guys? So this you need to know and see this. It is non-toxigenic negative control and it is not producing any sort of percipient line. So this is used for the toxin demonstration. Is it fine? And regarding diphtheria, I like to tell you one or two important points, guys. And those important points are this way, that every CD is not pathogenic. It is made pathogenic by beta-phage tox gene. And this conversion is termed as what? Lysogenic conversion. And lysogenic conversion is an example of transduction. And whenever you heard of this transduction, what should come in your mind? This particular bacteriophage or phage, whatever you call it. Okay, this was that Fmge question only last to last time. Bacteriophage mediated transfer is termed as what? The answer is transduction. Okay, so this is what is the answer. And if we talk about the culture medias and all, early diagnosis, Loeffler serum slope, like definitive diagnosis, potassium tellurate agar. Okay, and this dictum you already know that it is having pseudomembrane, and when you try to plug it up, that bleeds. And this particular is having this bull neck and stuff and all that. Okay, so that you already know. Besides this, this dictum is also important. Diphtheria is dash, not dash. It's toxemia, not bacteremia. So that's why we are demonstrating the toxin here in this particular one. Okay, guys. So these all are important stuff regarding diphtheria. And along with that, I like to tell you regarding non-corny diphtheria also. And important one is Corynebacterium minutissimum, and that leads to erythrasma. Okay. And that is having coral red fluorescence. You must have read in the like skin also that particular thing. And we have made to remember it in this particular stuff. Like Corynebacterium minutissimum, Minu's mom prepared up red rasma, that is, or red rasam. So, and any sort of that erythrasma. So, important question. Very good, Dr. Anil. Albert, Abdul. Yeah, right, very right. Albert Stein. Albert Stein is there. Chinese letter pattern is famous for it. Green colored one if it is Albert. Otherwise, you are having this purple stuff. It is a gram-positive bacteria. It's a gram-positive bacilli. So it can take up both the stains. Okay. So this is regarding the diphtheria. Antidiphtheric serum. That is the like treatment of choice. Now, guys, coming to this one. Can you tell me what you are observing in this particular picture? Up. Tell me this. What you are observing in this? Is it a gram staining? Is it Zn staining? And if it is Zn staining of what we are observing this? Tell me this answer. Yeah, Hare Krishna, very right. Chinese letter, cuneiform arrangement. Yeah, tell. This particular one, please. Zn staining or gram staining? What it is, guys? Tell me. Zn staining or gram staining, first of all? Yes, anybody? Yes, please. The comment section. Yeah, SM, it's very useful for NEET PG also. Yeah, 24 SM. Don't miss it. C. The answer told by the students is C, and they are right. It's Zn staining and it is for the Mycobacterium TB. And you know that Zn staining, what is the steps of Zn staining? Carbonyl fission. And the mnemonic for that is car inside se hai blue, means carbonyl fission. Then you do the intermediate heating and then 20% H2SO4 in this particular case. And then you're using of what? Methylene blue. Okay, so background actually not the proper blue. It is not a proper stain. But these particular things, they are pinkish to reddish in color and they are arranged like that. We are typically talking about what? Zn staining. And for the gram staining, the mnemonic is what, guys? Methyl gas. So that also you need to know. Methyl violet, okay, grams iodine, acetone and safranin. Okay, gram positive bacteria, that is they look like purple to violet, and gram negative pink. Why? The most important factor which governs the gram staining is peptidoglycan thickness. And peptidoglycan thickness there in the cell wall is whom? Gram positive or more thick. More thick cell wall is the feature of gram positive bacteria. Okay. The question which have come recently is in your exam only in the Fmge, this was there that lipopolysaccharide, that is a feature of which cell wall first of all? The answer is gram negative. Okay. And from that there is an antigen which comes out that is termed as O antigen. Now what is O antigen? Somatic antigen. Okay. O antigen is what, guys? O antigen is somatic antigen. Okay. Dr. Anil, you have done the research, huh? Are you a microbiologist? Slow growing, chemo, organotropic, non-motile, non-spore forming aerobic bacillus. Oh, bhai. Good. Yeah, very right, Abdul. Very right, Vikas. Negative, what negative? What negative? Preeti, what negative? Answer is C. Okay. What is full form of ZN, guys? Ziehl-Neelsen. Okay. And who is the actual discoverer of this particular acid-fast staining? That is Paul Ehrlich. Yeah, very right. O antigen is somatic one. Now, guys, tell me this: the famous culture media, which you should know. Without knowing this, you should not go in the exam. Trick से याद किया हुआ है. हो. लेकिन यार उसकी ज़रूरत नहीं है अनिल. इतना याद करने की ज़रूरत ही नहीं है भाई. No need. Actually, प्रॉब्लम पता है कहाँ है? प्रॉब्लम ये है कि वी शुड ऑल्सो नो दिस कि व्हाट शुड नॉट बी लर्न. यू आर नॉट सिटिंग इन द एमडी एग्जाम यार. तो वाई आर नीड टू रिमेंबर एवरीथिंग, कीमो, ऑर्गेनोट्रॉपिक, स्लो ग्रोइंग एंड ऑल दैट. So no need, I suppose. But as such, if you are able to remember it, good. No problem. Yeah, tell me, which particular media, guys? Without this, you should not go in your exam. NEET, PG, INICT or any. Which particular one? Vikas have told wonderfully, yes. The answer is LJ. LJ stands for what? What is the LJ full form? Lowenstein-Jensen media. Lowenstein-Jensen media. This is having how much percent agar? Every particular media is having agar, na guys? 2%. It is having how much? LJ media is not having agar. Okay, it is having eggs, which play the role of solidifying agent. Okay, so that you first of all know. And because of that only, we inspissate it. Inspissation is being done for these LJ media, not the autoclave. Otherwise, for most of the culture media, what you do? Autoclaving. For this, you do what? Inspissation. Both are moist heat sterilization. Rough, tough and buff. Preeti, you're right here. Buff, rough, tough and buff. And how much time the colonies of this particular Mycobacterium TB comes on the Lowenstein-Jensen media? Can you tell me? No agar. Daffodil have come with no agar. Yes, you are right, daffodil. Daffodil used to be my house in my school. The flower. Okay. Yeah, rough, tough and buff colonies, they are present up 6 to 8 weeks it takes to them to come on this particular one, and it is a sort of eugonic or dysgonic growth. The answer is eugonic growth, luxuriant growth, but it takes time to come. Now, here only I like to tell you regarding these C and D options. Okay, so Dorset egg media is also used for TB only. Nobody knows it, but it is true. Dorset egg is also used for what? Mycobacterium TB, and Pitra greeni media is also for the TB. Okay, Mycobacterium TB. But this you already know, Loeffler serum slope is for what, guys? It is for Corynebacterium diphtheria. Okay, it is for Corynebacterium diphtheria. Okay जी. तो मैंने बता दिया, like culture media mostly autoclaved, but this particular one is what? This particular one is inspissated. And inspissation and autoclaving both are what? Moist heat sterilization. They are what? They are moist heat sterilization. Now guys, this particular table itself is full of questions. Okay, this is full of questions, and short, this particular question is coming regarding the E. coli pathogenesis. Okay, so let me tell you regarding the important points here. Okay, I will just select up the important points. Yeah, Kartavya, you're right. See this particular one, guys. First of all, see this Fimbria. This was a NEET PG question also. Fimbria helps in what? The Fimbria helps in adhesion. First point. Okay. And this particular P. Fimbria, this is there in certain E. coli's which attaches to the uroepithelial cells. And they leads to what? They leads to UTI. Okay. That leads to what? That leads to UTI. And this assists in what? Urinary colonization, that you already know. So P. Fimbria, they helps in adhesion. Now talking about pathogenic, enteropathogenic E. coli. So guys, P for P, that is pediatric age group diarrhea. It causes diarrhea in the nurseries. That is one of the points. Another important point regarding this particular fellow is that it leads to A/E lesions. And what is A/E lesion, guys? attaching. It attaches to the intestinal alveoli and damages them, effaces them. So that is termed as A/E lesions. That is done by enteropathogenic E. coli. Okay. Then you see this side, guys. CFA, colonization factor antigen. You're observing this colonization factor antigen, and this particular CFA is present in ETEC, enterotoxigenic E. coli. This question can come this time, and when I say this question can come, usually it comes. Okay, so CFA is colonization factor antigen, and this is present in what? This is present in ETEC, and ETEC is toxigenic E. coli, which is one of the common causes of what, guys? Traveler's diarrhea. Which is the common causes of what, guys? Traveler's diarrhea. It is the most common cause of that, traveler's diarrhea. Which one? ETEC. And you will be having an interesting aspect here, that this is the most common cause of diarrhea in children if bacteria is asked. Pediatric is fine, but overall in the children, if somebody asks you, as per the bacteria, ETEC. But overall, obviously, you know, rotavirus is there. But bacteria, if it is asked, then it is ETEC. Abdul have told, right, travelers. Now, guys, these are the toxins, ST toxins and LT toxins of it. That is stable toxin and labile toxin of the ETEC, which causes diarrhea. And stable toxin, I already told you in my lectures. Stable toxin acts through which mechanism, guys? Increase in cyclic GMP, guanosine monophosphate. Okay, stable में कौन रहता है? In the stable, what resides? Horse. And what you call horse in Hindi? Ghoda. Okay, increased cyclic GMP is for the stable toxin. And this particular same thing goes where also? Bacillus cereus. Bacillus cereus hematic strain, Bacillus cereus diarrheal strain. Yes or no? Manisha, very right. Rota, you have told. And travelers, you have told. Okay, so increased cyclic GMP is stable toxin. Labile toxin is increased cyclic AMP. That is adenosine monophosphate. So these two things you need to know. Now, virotoxins. Another question which is there likely coming in the exam is this. Virotoxin associates with HUS, guys, and the bloody diarrhea. And which particular one is there? EHEC. Okay, as it written also. Virocytotoxin or virotoxin, also termed as Shiga-like toxin. Okay, it is also termed as what, guys? Shiga-like toxin. Fine. So this is also there. Besides that, what is the culture media? The culture media for this particular is SMAC. And what is SMAC? Sorbitol McConkey agar. Okay, sorbitol McConkey agar is the culture media. This was asked in NEET PG this time. And the next question might be that which other media is used? The answer is rainbow agar. So SMAC and the rainbow agar, that is there in this particular one, which you need to know. Okay, and as you can see that certain E. coli, they having capsule also, certain ones, not everybody, certain ones they having. And as we have talked about the lipopolysaccharides, you know, lipopolysaccharides, they lead to the release of endotoxins, and endotoxins leads to what? Endotoxic shock. So that is also there. And those who have this capsule and this K1 antigen, the capsular antigen, they enters into brain and they leads to this neonatal meningitis also. And this is explained up that these are interleukins which are released up and the patient lands in what? Endotoxic shock. So this particular flow chart itself is telling up a lot of questions regarding the E. coli, and we are talking about the diarrhegenic E. coli mostly, and here we have talked about the UTI also. Okay, here we have talked about the UTI also, and one of the questions NEET PG was an INCT. What are the screening tests for the UTI? Screening test for UTI is grease nitrite test, okay, then leukocyte esterase test, and catalase test. These are the screening tests, and obviously definitive and confirmatory is culture. And culture is done on which media, guys? CLED. And what is CLED? Cysteine lactose electrolyte deficient. Okay, why not McConkey agar? Because McConkey agar is selective, CLED is non-selective. Okay. Very good, very good, very good, very good. Namaskar, namaskar, namaskar. Chale aage? So I suppose you must be knowing up a lot of things now. Now, this particular one, guys. Choose the right statement on the basis of given media, and as you can see this picture, if I zoom it up, so these are jet colored colonies, and jet colored colonies, they are of Salmonella typhi on which media, guys? On which media we have this jet black colonies? So jet black colored colonies of Salmonella typhi on which media? Wilson and Blair. Wilson and Blair. Wilson and Blair is having jet black colonies of Salmonella typhi, and this is what is the picture of this particular one, which you should know. Okay. Otherwise, if we talk about the various culture medias here, the transport media is SACS. This was asked. Okay. Enrichment media is selenite broth, and the best selecting media is Wilson and Blair. And the same medias exactly they are used in the Shigella, but Wilson and Blair is exclusive of what? Salmonella typhi. Okay. And the Salmonella typhi have come here. So let's talk about Salmonella typhi also. The question of last FMG was that in which particular week, which particular test is done. So first week, second week, third week and fourth week. And remember, this is week. And they have asked you on the third day, which is the best test to be done. So obviously on the third day, it is the blood culture, which is best. And there is bone marrow culture, which can be done if you have that particular capacity also. Otherwise blood culture. And the question asked was third day. And third day obviously comes in the first week. The answer of R should be what? Blood culture, but everybody have written stool because they were not knowing that this is week. Okay. And the question asked regarding what? The question asked regarding the day. Okay. So A is agglutination, and agglutination is which test? That is the Vidal test, which nobody does it. Now they go for typhoid hot and all. Okay. S is stool, that is stool culture. U is urine culture, which is actually not done proper. ELISA, that is being done, urine ELISA, along with the urine culture also in the fourth week. Okay. Yeah, very right, very right. Very good. So this is what is the mnemonic for this Basu. And another question which can come here is this particular Fmge sign. And Fmge sign is relative bradycardia, you know. Okay. Step ladder pattern, rose spots, okay, pea soup diarrhea. They all are features of what? Salmonella typhi typhoid fever. And what is the incubation period, guys? This particular incubation period is definitely 1 or 2 questions in NEET PG as well as in the FMG exam. And this on my YouTube channel by my name only, I have made a video of this. So you can see that incubation period 1. Definitely 1 or 2 questions might be coming from that. I will share the link. Okay. So this is there, guys, and important is that in this, the leukocyte count does not increase as other fevers. It remains normal or it goes down. So this is also a characteristic point of typhoid fever. Okay. And the culture media is already talked about, and drug of choice you already know, that is the ceftriaxone. Okay. Yeah, Preeti, very right. Now tell me, guys, about this particular picture. Greenish color picture, and antibiotics are seeing in this. What we are talking about and which media it is. Yes, please tell. Manisha, very right. This question is coming. Manisha, I was telling na, osteomyelitis in sickle cell anemic patients. Yes, Salmonella. Salmonella, certain species, they can definitely does the osteomyelitis. They are the most common cause of osteomyelitis in sickle cell anemic patient. And this question is coming. Manisha, I've told it wonderfully. Yes. Okay. So this particular antibiotic testing is being done for the Pseudomonas, which have turned this MHA plate also green because of the pigment pyoverdin. Okay. And now if we are talking about Pseudomonas, in the Pseudomonas, the first question which comes up is that what is the culture media? The culture media is citramide agar. Which particular important conditions associates? Important is malignant otitis externa. Common in whom? In the DM fellows. Important here, ceftriaxone and vancomycin is not anti-pseudomonal, first point. Okay. And polymyxin B and colistin. In fact, polymyxin B only is remaining now, against which no resistance is reported up till. Okay. So that was also being asked. Burkholderia malae, Pseudomalae, Burkholder capacea, they are the brothers of Pseudomonas. They come in the family of non-fermenters. Okay, Pseudomonas is what? It's catalase positive as well as oxidase positive. But one guy which is nowadays having a swag, that is Acinetobacter baumannii. That is oxidase negative. And it is found where? In the ICU. And it is multi-drug resistant isolate. Okay, so these are the important ones. Yeah, very right, Abdul. Burn wound. The favorite of that particular one is burn wound. And Arvind is telling up this is KBDDT. Very right, guys. This is Kirby-Boyer disk diffusion testing on MHA. And if MHA is not there in the option, the second best answer is Na. That is the nutrient agar. Is it fine, guys? Swarming is a feature of Proteus. Naglers is there in perfringens. Dynes phenomena is there in Proteus. Okay, P for P. Now, this just now we have talked about oxidase 1. And oxidase is actually what I should tell only. Okay, oxidase is this white color changes to blue. This white color, the strip changes to what? Blue within 10 seconds. Okay, this particular one changes in blue within 10 seconds. And this is what is termed as oxidase positive. And the bacteria which have just gone, it is strongly oxidase positive and catalase positive both. Okay, catalase and oxidase positive both. So answer to this question, can you tell me except? So guys, this is slightly tricky question because you need to know a lot of things regarding this. But I should tell you one thing, if you are smart enough to exclude other things, then you can easily exclude up that E. coli. E. coli is an Enterobacteriaceae member. Okay, and Enterobacteriaceae family is fully oxidase negative. Okay, so answer of ours is obviously E. coli, and smartly you have reached to that because Brucella is oxidase positive, Vibrio is oxidase positive, and Neisseria both, gonococci, meningococci, they both are oxidase positive. Okay, they both are oxidase positive. Yeah, very right, very right. So answer is D, E. coli. Okay, and this is the oxidase test. Now guys, if you see this particular picture with zoom in, you can see that in between there is no staining and the poles are stained. So which particular staining we are talking about, in which the poles are stained and in between it remains unstained? What we are talking about, guys? Kartavya, it's better to remember this particular thing, that which are oxidase positive. Okay, because this word is oxidase negative. Yeah, Preeti, you are very right. Answer is Yersinia only. But what is the answer? A, B, C, D. Which is the answer here? A, B, C, D. Bipolar staining. Answer is, yes, answer is, answer is C, Giemsa stain. So you can stain the Yersinia pestis by the Giemsa also, and this have come in this NEET PG exam, Giemsa stained. And if it is for Giemsa stain, one more stain name you should know, Wason stain. Wason staining and Giemsa staining, the characteristic done for Yersinia pestis and this particular Yersinia pestis. Can you tell me the culture media for this? What is the culture media for this? What is the culture media for Yersinia pestis? Yeah, in the comment section. What is the culture media for Yersinia pestis? Ghee broth. Okay, what is the culture media? Ghee broth, and stalactite type of growth is there, and there are three types: bubonic, septicemic and pneumonic. Okay, and in this, mostly the question now they come in the form of this, that somebody have gone to the foot, go down, and there is darkness, somebody have bitten up, and there is a swelling in the inguinal region, inguinal buboes, and when you go for this inguinal buboes smear, biopsy smear, you observe this safety pin appearance. Okay, so we are talking about this particular stuff, that is the Yersinia pestis. Yersinia enterocolitica. This is the new one which can be asked in the NEET PG and ICT also along with Fmge exam. It shows appendicitis-like features. Okay, it shows appendicitis-like features, and important aspect is differential motility is observed by the Yersinia enterocolitica. Okay. Now coming to this particular one, another important question which can definitely be asked in the exam, and as it is written up, Haemophilus influenzae colonies are there and Staph aureus is being streaked off. If these two names comes, immediately what should come in your mind is satellitism. And this was again asked in the NEET PG. Satellitism is shown by which particular bacteria? The answer is Haemophilus influenzae shows it in the presence of Staph aureus. And satellitism is actually the presence of certain factors X and V near the streak of Staph aureus. That is what is termed as satellitism. Is it fine, guys? So satellitism is this particular entity, and this was asked by this name only, that we are talking about which particular organism? Haemophilus influenzae. Haemophilus influenzae is a gram-negative cocobacillus, first thing, loves to grow on the chocolate agar. The capsulated bacteria, they are more invasive, leads to what? Meningitis, epiglottitis, normal ones, LRTIs, okay, otitis media, they are being done by what? Non-capsulated one. So these you need to remember in this particular sense. Besides that, satellitism is shown by this. It is catalase and oxidase positive, okay. So these all, they can be asked in some form or another regarding it, okay. So answer is B, as everybody have told up. Now guys, this one, this everybody should see and tell that what culture media we are talking about and which colonies are there on this particular one. Then we'll talk about certain important points regarding it. Yes, can you tell me guys, which particular media? Yeah, Anil, tell me Vikas, tell me Daffodil, tell me Krishna, Cortex, Preeti, tell me. Which culture media it is? By seeing up only, if you are telling up, what is the answer? Is it XLD? Is it TCBS? Is it DCA? Is it CLED? What it is? And the Medico Journey is telling D. Arvind Kumar is telling B. Arvind Kumar again is telling TCBS. So guys, tricky one. Again, this is a tricky one. As such, what I want to ask up is this only, TCBS, thiosulfate citrate bile salt sucrose, which is a culture media, best selective media for Vibrio cholerae, and it is a greenish color media on which this particular yellow stuff, yellow colonies are coming up of the Vibrio cholerae. But you are also right guys, this can be CLED also. But what is the clause for that? It is not showing the growth of Proteus, because Proteus is NLF. It is a non-lactose fermenter. It will not change the color to yellow. But yes, if instead of Proteus, what is being written off? E. coli, okay, Klebsiella, which are lactose fermenters, Shigella soni, they will change the color like this. Then the question will become tricky one. Okay, but as such, the answer is TCBS here. And CLED, cysteine lactose electrolyte deficient, this is not the answer because we have told up regarding the Proteus, which is a non-lactose fermenter. But remember this also, CLED is also green color and it changes to yellow when the lactose fermenters grows on it. Remember that. Which one is better, CLED or McConkey? CLED is better because it is non-selective. Another NEET PG question. Okay, DCA, deoxycholate agar is used for Salmonella and Shigella. It is not of this color. Okay, it is not of this color. Now, as you can see this particular picture, the lesion is painful, bleeds easily while taking the swab for the gram staining. What is the probable cause after seeing of this particular picture? Is it Haemophilus ducreyi? Is it Trypanosoma pallidum? It is HPV or HSV2? Yeah, Dr. Anil, you are right. Rice watery stool, which mimics of which particular poisoning? Arsenic poisoning. And the question which have come recently is that the cholera toxin acts on tight junctions or zonooccludins. They are the same thing. Okay. And in that the B is there, the B subunit. It attaches to which particular receptors? GM1 ganglioside receptors, and it does ADP ribosylation of G stimulatory protein. That you need to know. And there is increase in what? Cyclic AMP. And you are right guys, the answer is Haemophilus ducreyi. Haemophilus ducreyi, dukh raha hai, it's painful and it bleeds. That is what is chancroid. And the point which I want to tell up here, when you take the biopsy from here and stain it with Giemsa, you will find what? School of fish. School of fish appearance or tram track appearance. That is observed in this particular one. Okay. And Trypanosoma pallidum, the same thing, is painless, and this particular painless stuff, along with that, it is a hard chancre over the genitals. Okay. In this, it is a soft sore, soft ulcer, which is painful. Krishna, you are telling dukh raha hai to chancroid. Yes, you are right. Now, This I have already told up. This is a repetition one. I have already told up this one. Ghee broth. Which particular organism loves to grow in ghee broth? So do you know? Answer is Yersinia pestis. Okay. And the important point regarding Acinetobacter which I have told you, it is oxidase positive. Sorry, this oxidase negative. And this particular Acinetobacter, why it is famous? Because it is found to be a MDR isolate in the ICU. Okay. MDR isolate where? In the ICU. बिल्कुल इंपॉर्टेंट है डॉक्टर अनिल, इंस्टाग्राम के क्वेश्चन पोस्ट बिल्कुल इंपॉर्टेंट है, वो देखो सारे के सारे. हाँ, वेरी राइट. आंसर इज Yersinia pestis and Acinetobacter you need to know is oxidase negative and MDR isolate है in the ICU. And recent INI-CD question, this name at least you should know, Stenotrophomonas multifila. By chance, if you get up this particular name in your paper, which you might get, Stenotrophomonas multifila, this is also a non-fermenter. Okay, this is also a non-fermenter. Interestingly resistant to number of drugs and susceptible to cotrimoxazole. This was asked in INI-CD and this can be repeated again. Okay? Okay guys? Let's move. Now, another favorite question. Everywhere darkness is there and against that you are observing these particular fellows moving. So we are talking about what guys? Dark field microscopy for the Treponema pallidum. And Treponema pallidum is a spirochete. Okay, so this everybody knows it, this particular thing, dark field microscopy. And in the dark field microscopy, the question comes, which particular light you use? Answer is reflected light. Okay, and in the normal microscope, what we use? Transmitted light. Okay, so this you need to know. The dark field microscopy is using up the reflected light and it is used for the Treponema pallidum. Fine, and they are spirochete, that's why they look like that, spiral in shape. Fine. And the question recently in the INI-CT was syphilis incubation period, that is 9 to 90 days. What do you get in the primary syphilis? Painless heart chancre. What do you get in secondary syphilis? Rashes over palms and soles. Along with that, condylomata lata. Then comes the latency. Then it goes into tertiary one. Then it is having gummas on the skin. Besides that, along with that, you can have those neurosyphilis and cardiovascular syphilis features. Neurosyphilis stapes dorsalis. Okay. Cardiovascular aneurysm of aorta, ascending aneurysm. What is drug of choice? Penicillin, drug of choice. Okay. And what is the best test as such, most sensitive if I ask you? The answer is FT-ABS. Most specific, TPI. For the prognosis of syphilis, that is VDRL. VDRL, Venereal Disease Research Lab. This particular test, Venereal Disease Research Lab, this is which type of a test? It is a slide flocculation test. What is flocculation? Flocculation is a type of precipitation. Okay. Precipitation, what is the difference? The precipitation, you use a soluble antigen with antibody. Okay, so this particular soluble antigen is used in that and there is a precipitate formed. So instead of precipitate, in the case of VDRL, what is produced? Floccules. Okay. Very right, 9 to 90 days, very right, wonderful. Now, tell me about this. Which bacterial colonies they look like this particular one? So this is the fried egg appearance of which particular bacteria? This is fried egg appearance of which particular bacteria, guys? Can you tell me? Fried egg appearance of which particular bacteria? Yeah, tell. Fried egg appearance of which particular bacteria? Right, Manisha, you are right. The crystalline penicillin also penicillin only, na? Which bacterial colonies appears on the solid media? These particular tab. The answer is C, Mycoplasma pneumoniae. Dr. Naveen, you have taken the lead. You are right. Dolly also have told up wonderfully Mycoplasma. The answer is Mycoplasma. And Mycoplasma produces fried egg colonies. Now the winner will be those who will tell up that which other guy does produce fried egg colonies same like this. If anybody tells that, that fellow is scoring here itself about 200. I am telling off. Which other organism produces fried egg colonies? Let me give you a hint. That's a fungus. That's a fungus. Tell me. That's the fungus. What is the answer? which produces fried egg colonies like Mycoplasma pneumoniae? Fast, tell me. Oligodendroglioma, पूजा ये क्या है? वो क्यों बनाएगा फ्राइड एग कॉलोनीज? Shivangi Tiwari, you are perfectly right. The answer is Malassezia furfur. Okay, which leads to Tinea versicolor or Pityriasis versicolor. It produces fried egg colonies on the SDA. Okay, on the SDA. And if you are talking about Malassezia furfur only, then you should know spaghetti and meatball appearance, which was the last Fmge and NEET PG question. Spaghetti and meatball appearance is observed when you put those scales into 10% KOH and see them under microscope. You could able to see those particular short hyphae. Okay, and that is what is termed as spaghetti and meatball appearance. Modi ji, आप बताएं. चिकन वायर की तो बात ही नहीं हुई भाई, फ्राइड एग कॉलोनीज की बात हो रही है. चलें आगे. चलो इनके बारे में बात कर लेते हैं भाई. कॉक्सिला क्या करता है? कॉक्सिला बर्नेटी लीड्स टू क्यू फीवर, क्वेरी फीवर, फाइन. एंड दिस पर्टिकुलर वन कम्स इन द रिकेट्सियल फैमिली, दैट you know. And Coxiella burnetii is an organism which leads to culture negative endocarditis, fine. And another important thing regarding Coxiella is that it does not produce any rash. It produces pneumonia-like features. It is the atypical type of pneumonia, and this particular one associates with the cattle birth process and all that. So that you should look out for the history of the question. ये क्या लिखा भाई? आपका आप है किस सर? अच्छा, इंडिविजुअल ऐप आपका. इंडिविजुअल तो नहीं है भाई. दैट इज ऑन द एलन नेक्स्ट नाउ. यू कैन हैव फ्रॉम देयर. द केयूर वधार. राधे राधे, राधे राधे जी. Bordetella pertussis leads to pertussis, also termed as whooping cough, okay, and also termed as barking dog cough. Incubation period here is also 7 to 14. Typhoid is also having the same 7 to 14. Catarrhal, paroxysmal, convulsant, these are the three phases. Borded ganju media is there, bisected pearl appearance, and the drug of choice was asked in the Fmge exam. Answer is azithromycin in the case of children. Okay, so these are the important ones which you should know. This is also important. Legionella. Legionella, guys. Legionella. Can anybody tell me what is the culture media for this? The culture media for this is BCYE. What is the full form of BCYE? Buffered charcoal yeast extract. Okay, buffered charcoal yeast extract. Moderate ganju media. It is having bisected pearl appearance. BCYE is the culture media for Legionella, and again Arvind is telling us what is drug of choice. Legionella also azithromycin. Okay, so कहीं कुछ समझ में ना आए ना तो what should you put the answer? Azithromycin. Two drugs are very famous. One is Azi and one is Doxy. Out of them you can do akkad and you can put up. For this also the Azi is the answer. Yeah, very right. Pushpendra, pneumonia, diarrhea. This particular triad is termed as Legionnaires disease, and this is the pneumonia in which what you have electrolyte imbalances like hyponatremia and all. So that also you need to know. Those who are actively participating now, you all are passing, man, this time. And let me tell you, those who does microbiology na properly, they are the persons who pass up with a wonderful marks, because they don't know that as such only it's told up that 13-14 questions they come, and if you see of the proper one na, it links to 30 to 40 questions, and 30-40 questions does matter, man. Okay, chalo आगे चलते हैं. Another favorite of examiner is this particular thing in which you have sufficient clue, that is clue cells. And clue cells is a feature of which particular disease, guys? Clue cells. This is a clue cell here also written up. Can you tell me what should be the answer of this question, guys? Yes. Yes, yes, yes. What should be the answer of this? Clue cells. Thank you, Delirium. Now I am on Ellen next. And everybody telling wonderfully well. Answer is Gardnerella vaginalis, and Amsel's criteria is there. The pH goes more than 4.5, and you can able to see the clue cells here. And when the discharge is the greyish white one, that is fishy only, fishy smell. And when you put a 10% KOH in that whiff test, it enhances that fishy smell. So that is what is Amsel's criteria. And drug of choice, Kayur have already told up, metronidazole. Okay, and in pregnancy also, Kayur, answer is metronidazole. Favorite question examiner must have suffered from bacterial vaginosis. Pushpendra have written everything right. Very right. Good. Good, 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 good. That criteria, you know, Amsel's criteria. Can you tell me another criteria which was used to be used before? What was that? What was that? Anybody? And if you answer that again, 200 above. Pawan, wonderfully, yes. Nugent score. You are right. You are right. पवन तुम्हारे आ रहे हो भैया 200 से ऊपर इस बार, अगर FMG में हो तो, नहीं तो नीट PG में पास तो हो ही रहे तुम बढ़िया से. हाँ जी Delirium, you are right. In trichomoniasis also metronidazole. Strawberry cervix, strawberry vagina in trichomoniasis. Good, 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 guys. Okay, moving. Now, see this particular one. There is a formation of eschar at the site of mite bite. Identify the causative agent. Mite की बात हो रही है और causative agent पूछा है. Okay. So guys, we are talking about which particular disease? We are talking about scrub typhus. And scrub typhus is transmitted by mite and done by what? Aurantia sussugamusi. पुष्पेंद्र डेलिरियम ने बहुत ज़्यादा पढ़ लिया है. More than 7 they are telling for the Nugent. Nugent अब यूज़ ही नहीं करते, इतना कॉम्प्लेक्स है वो. Eschar at the site of mite bite, identify the causative organism. Scrub typhus is the disease and the organism is Aurantia sussugamusi. And in this, important is that if you are having in the history a black eschar, a rash, okay, and a hilly region history, these all features are there. Okay, they talk about this and they talk slightly regarding the Wheel Phalyx test. Wheel Phalyx test is OXK, strongly positive. Scrub, like this you remember, no? Scrub typhus. Ivermectin कहाँ से आ गया मनीषा? Ivermectin किसके लिए यूज़ करेंगे? हैं? अरे नहीं भाई, बुरेलिया कब से कुटीने लावा माइग्रेन्स करने लगे पवन? अरे डेली इस बार तो तुम पास हो यार, टेंशन मत लो बिल्कुल भी. फुल कॉन्फिडेंस से जाना, विद फुल कॉन्फिडेंस. OXK is strongly positive and Abdul, you are right, it is also termed as chiggerosis. Okay, chiggerosis. Wheel Phalyx test is which type of a test? First of all, tell me this. It is heterophile agglutination test. Which is the other example of heterophile agglutination test? Paul Bunnell test. Okay, tell me in the comment section, Paul Bunnell test is done for. बस KBs नहीं है भाई. इश्कार कहाँ मिलता है इस KBs में? Doxy is drug of choice. Yes, you are right. मैं बोला ना Doxy ऐसी, इन दोनों में से कोई भी गैस कर दो, आंसर तो होगा ही वो. Right? बताओ. Which other heterophile agglutination test you know? Anybody? If somebody tells up this, 180 marks this time. Tell me. Paul Bunel test. Preeti, you are passing this time. Okay, Paul Bunel test. Paul Bunel test is done for which particular virus? Infectious mononucleosis, yes. Which particular virus does it? Epstein-Barr virus. Okay, very good. Epstein-Barr virus is which number, guys? HHV4. Human herpes virus 4. Okay. So these are important ones which you should know, and as such, if I talk about the rickettsial family members, epidemic typhus, endemic typhus, Rocky Mountain spotted fever, Indian tick typhus, okay, rickettsial pox, these all are important diseases, and you should know regarding their causative organisms. Drug of choice for all of them is doxycycline, that is also there, okay, and in this rickettsial family only certain fellows they come like Bartonella Hanslet, which leads to cat scratch disease, which used to come before, but now they are telling that they're different in some characteristic. So Bartonella Hanslet is one of them, important. Cat scratch disease has been done by that. Stellate granulomas, done by the scratching of the cat. Haste the cat has scratched you. Bartonella quintana, that leads to trench fever. And both of them, they lead to what? Bacillary angiomatosis, which is a type of a vascular tumor like Kaposi sarcoma, and obviously common in whom? In HIV positive one. And one is Bartonella bacilliformis, which leads to Oroya fever. That also is there. Okay. Lithium causes it. अब भाई तू कैसी कैसी बातें कर रहा है, क्यों रहा है? Lithium क्या करने लग गया? Delirium, you are right. CMV choose for 5 years. And Epstein-Barr virus, that is HHV4. ये है, इतना इंपॉर्टेंट तो नहीं है, लेकिन हाँ बाई चांस पता लगा किस्मत ही खराब उस दिन, तो यू शुड नो एंडोफ्लैगेला ऑफ ट्रिपिनोमा पैलिडम. Trypanosoma pallidum is not having a true flagella, it is having an endoflagella which arises from the periplasmic space, and this Trypanosoma pallidum is having which type of motility, guys? Corkscrew motility. Okay, it is having a corkscrew motility. Now talking about fungus, some of the fungus we are talking about, and in this particular fungus, obviously these names suggest that we are talking about what? We are talking about dimorphic fungus. We are talking about dimorphic fungus. What dimorphic fungus? Dimorphic fungus exist in yeast as well as mold form. Mold is gold. How much carat gold? 25 carat gold. So 25 degrees centigrade. And yeast at which temperature? Body temperature 37. So that you already know. Okay. So this is a dimorphic one. And let me explain up the important points in dimorphic fungus. And if you like make it zoom one. So blastomyces. The important point regarding blastomyces infection, blastomycosis is it is having figure of 8 appearance of the yeast cell. It looks like a figure of 8 or broad based. Coccidioidozymitis. It is having this arthroconidal arrangement, and there is a spherule which have endospores inside. Histoplasma. It is having a tuberculate macroconidia. Remember guys, histoplasmosis is now a favorite of NBE. They can ask you again this with this particular feature, and this is termed as tuberculate macroconidia. Looks like a sunflower. Okay, and histoplasmosis, what they are asking is that chronic histoplasmosis mimics TB because cavity forms in that, and in TB also cavity is there. That is important point. And there is a narrow based budding like this. This is termed as narrow based budding in that. Okay, and these narrow based yeast cells, these budding yeast cells, you can find in the bone marrow biopsy, guys. In the bone marrow biopsy of these individuals with a traveler history, if they are coming up in your exam, they are asking regarding that, they are asking regarding the histoplasmosis, which is also termed as OYO disease. Okay, OYO disease, and this is also termed as Darling's disease. Yeah, everybody is telling in the comment section. Good, wonderful. Fine. So if they are talking about bone marrow biopsy and they are showing up the narrow based budding yeast cells, they are talking about histoplasmosis, one thing. And if they are talking about traveler history and they are talking about the bone marrow biopsy, and in that bone marrow biopsy they are talking about the kinetoplast, okay, and some nuclear stuff, and they are telling that the color is changing black, they are talking about Kala-Azar, a mastigoid form. Remember that thing also. Okay. Paracoccidiidose, this particular one, Paracoccidium brasilensis, it have Mickey Mouse appearance of the yeast cell. Mickey Mouse appearance, mariner's wheel appearance, that is typical of this Paracoccidiidose. Sporothrix kinki, sporotrichosis, rose gardener's disease. This is termed as flower-like sporulation, the mold form, and this is termed as narrow base budding. What we are talking about, guys, we are talking about dimorphic fungus. Okay, we are talking about what? Dimorphic fungus. Medical expert, पढ़ लो अब तुम, ठीक है? So these are what, guys? These are the dimorphic fungus. Fine. Now, talking about this particular thing, that zygomycetes. What is the other name of zygomycosis? Tell me that. What is the other name of zygomycosis? It is also termed as mucormycosis. Okay? Mucormycosis. Yeah, very right, Abdul. It is lymphatic spread. Sporotrichoid pattern is observed in that. Okay? And these particular features are there. Asteroid bodies. Wow. सब याद पड़ा है तुम्हें. Good. Good, good, good. Mucormycosis is the other name of zygomycosis, and the third name is also important. They have asked this time by that name, phycomycosis. Okay, phycomycosis is the third name of mucormycosis or zygomycosis. Septate hyphae or aseptate hyphae? Aseptate hyphae, obtuse angled. Okay, aseptate hyphae, obtuse angled. Fine. And obviously mucor and all, it's common in DM fellows, diabetes mellitus fellows and all that you already know, and COVID-19 history they can give you, and after that steroid use and all that, and you should know regarding it. So this is having aseptate hyphae, obtuse angle or right angle stuff. Pheoid, if this particular name comes, na guide, pheoid and all, this is termed as pigmented fungus. This name also can come in the exam, pigmented fungus, okay, darkly pigmented ones. And they are termed as melanized fungus. And these pheoid only have certain names which are termed as black fungus. So remember this particular point for your lifetime. Black fungus is not mucor. Okay, black fungus is pheoid fungus, and pheoid fungus are darkly pigmented ones. Let me tell you an example of this: chromoblastomycosis, which we're talking about now. So in which you have those medullary bodies and all that. That actually, that family is having certain fungus which are black in color. So that's why they are termed as what? Black fungus. Curvularia. You're right, guys. Ajay, very right. The drug of choice for that is often asked question, LAMP. That is liposomal amphotericin B. Okay, guys, let's move. This, we're just talking about the pheoid, na? This is that pheoid one. Melanized. If this word comes, a melanized one. So we are talking about the pheoid fungus. Okay, chromoblastomycosis we are talking about, and this particular appearance is termed as what, guys? Copper penny appearance. Copper penny appearance. What is the other name of it? Medullary bodies, sclerotic bodies. Okay, and the mnemonic for that is what? MSC. That is medullary bodies, sclerotic bodies, and copper penny appearance. Favorite question examiner. Must have suffered from this particular disease. It will become favorite of that particular fellow, and they are asking up again and again this particular one. Okay. Now, which fungal infection is this? Which fungal infection is this? Yes. This was also asked in the last exam. This is strawberry-like. Yes. Cortex sclerotic body is a favorite question of examiner. Must have suffered from it. What is it? This is polyps. Okay, strawberry polyps, nasal polyps, unilateral, bilateral, both. And it is rhinosporidiosis done by Rhinosporidium seaberry. Okay. And what you get in the biopsy of it? A spherule within that endospores. Drug of choice, somebody is telling up in the exam, like in the comment section, Dapsone. Manisha also told that it is non-cultured. Yes, all these points can be asked for sure. Okay, the chat is also like informative. Good. Now, tell me about this one. What is this particular test all about? Sean test. Which particular test we are talking about in this? Aquatic protozoa. Very good. This is aquatic protozoa, not the fungus. Yes. Which particular test we are talking about here, guys? This one. This is germ tube testing, also termed as Reynolds Broad phenomena. And it is being shown by which fellow? Candida albicans. It is shown by which fellow? Candida albicans. Germ tube testing. It produces germ tube formation. Okay, and along with that germ tube formation, it also produces these types of spores, which are termed as chlamydospores. And chlamydospores are produced on a nutrient deficient media termed as corn meal agar. Okay, and here I like to ask you a question. Beta-1,3 glucan testing. This particular beta-1,3 glucan testing, what was the mnemonic of it, and what is there in this particular cap? In this cap, you have this candidiasis, okay, then you have this aspergillosis, and then you have this pneumocystis xerovisi. It is not done for the cryptococcus, it is not done for the mucormycosis, it is done for the cap. Okay, it is done for the cap, and important is these are invasive ones. Invasive candidiasis, invasive aspergillosis, and this pneumocystis xerovisi. This is also a favorite of examiner, must have suffered from any of these diseases, and this question is a frequently asked one. Now, what is the drug of choice for the invasive aspergillosis, guys? Answer is voriconazole. What is the drug of choice for candidiasis? As such, azoles, that is the fluconazole and all. And if that thing is resistant sort, then you can go for amphotericin B and all. Very right, wonderful Delirium, wonderful Preksha, wonderful Manisha, wonderful Pushpendra. You are all rocking. Now tell me, which particular staining is this? The background is dark, and against that, this capsule is shining off. What we are talking about, guys? Yeah, tell this one. What is the staining this known as? Which type of staining it is? Tell. Which type of staining it is? Negative staining. Very right, Manisha. Very right, Daffodil. It is Indian ink staining. Indian ink staining is a negative staining in which the background is stained, and against that you can see this particular thing as capsule, and this capsule is virulent, and because of that virulence only it could enter into the meninges in the HIV positive ones. Most common cause of meningitis is Cryptococcus neoformans, and we are talking about that only. Okay, and for that latex card agglutination test is also available, which is better than this. This have also come because in that you can find out what? Cryptococcal antigen. And what's the drug of choice for cryptococcal meningitis? LAMP, along with flucytosine. Okay, along with flucytosine. Can we do beta-1,3 glucan testing here? No. For crypto, we can't do. Okay. Now this one, the favorite line, dichotomous branching of hyphae. That is a typical of which particular fungus? The answer is obviously Aspergillus, and it is having septations, and this is a sort of dichotomous branching at an angle of 45 or at an angle of 60 degree. That is what is the feature of this. No, stain, Pushpendra, that is not a stain, that is a test. That is the card test, latex card agglutination. Aspergillus is the answer, dichotomous branching of hyphae, and Aspergillus is having septate hyphae, dichotomous branching. The angle is acute angled, obviously. Dichotomous means that only 45 or 60. And important is those names: Aspergillus fumigators, Aspergillus flavus. Flavus secretes aflatoxin. Aflatoxin responsible hepatocellular carcinoma, found in groundnut workers also, that particular thing. And Aspergillus niger does otomycosis, blackish crust, and those particular aspergilloma, ABPA that you already know. Okay. And invasive we have just talked about. Invasive aspergillosis, drug of choice is voriconazole, and for that we go for the beta-1,3 glucan testing, and one more name, galactomannan testing. Galactomannan testing is also being done for that particular aspergillus. Okay. Now guys, this is a hot favorite of examiner. This might be seeing up in the exam. If they are showing up this, that is the crushed ping pong ball appearance, which are black colored, and the stain name is Gomori methamin silver stain, and obviously written up there the states of what Pneumocystis cerevisiae. This question might come in your exam. Okay, and in this you have perihilar opacities on the X-ray, and lobes are not involved. If at all they are involved, upper lobes are involved up in this. Pneumocystis cerevisiae pneumonia happens to be in HIV positive at the CD4 count of less than 200, and drug of choice for this is cotrimoxazole. This is also not cultured on SDA. That's why we don't consider it also as a fungus. Okay, it shows features of both, but still we don't consider it because it is not cultured on SDA. And this name or the stain is important, Gomori methamin silver. As such, it stains G for G, green, but the cyst stains black. Cyst stains black because cyst is having what? Glycogen. Okay. Right. Now, this is, I think, can come in your exam, and it can be INI-CT question also. We are talking about this reddish color stuff on this particular culture plate. We are talking about this Telaromyces marnifi. Telaromyces marnifi, before it was termed as Penicillium marnifi, and it is producing brick red color pigment. It produces what? Brick red color pigment, and brush border conidal arrangement is observed in this particular dimorphic fungus. In this particular dimorphic fungus, this particular thing is observed. And if they're talking about pigment, na guys, I should tell you this also, the Trichophyton rubrum, which is a dermatophyte, this is also having a red color pigment. Trichophyton rubrum, this can also come. And Trichophyton mentigrophytes, which is there na, in the dermatophytes. That particular fellow is hair perforation test positive, okay, and urease positive. So these are the important ones which I like to tell you. These are the important pigments. As such, there is no specific ones. Otherwise, black and all that, fuoid and all, they are produced in number of other medias also, and SDA also. Okay, but you remember two at least. Brick red on this particular SDF or Telaromyces marnifi, and another one which is producing this particular type of reddish color pigment, that is the rubrum. And how you can discuss that? Rubbing causes what? Redness. So flavus, aflatoxin release, Malassezia furfur, Taenia versicolor, mucormycosis, that you already know. That is a bacteria, na? Serratia marcescens. That produces red pigment, no doubt. This can also come red, right? You are Serratia, red, double R. Now this parasite, everybody knows about this parasite. It is having falling leaf-like motility. Can you tell me the answer of this, guys? Identify the parasite. This particular parasite, everybody should know. And if you don't know, then you should know it. Okay, otherwise it's difficult for the exam. Answer is? B कौन बोल रहा है? अनिल B कैसे हो गया आंसर भाई? How does the B answer? B पुराने वाले का आंसर अच्छा. I am asking this particular one. What is the answer of this? Answer of this is Giardia. Giardia lamblia. Giardia lamblia is Giradia. Which type of motility? Falling leaf-like motility is observed in Giardia lamblia. It is having this particular shape. These are eyes of it, that nuclei. In between there is an exostyle, 4 pairs of flagella. Falling leaf-like motility is observed in this. Okay, and the question which have come in your Fmge exam last time is Trichomonas. Name a protozoa which does not have a cystic stage. Answer is Trichomonas. No cystic stage is seen in this. Important. Naegleria fowleri. Naegleria fowleri is a brain-eating amoeba which leads to a particular ailment termed as PAM. Antamoeba histolytica. Antamoeba histolytica as such causes amoebiasis, intestinal amoebiasis, and it also does EIA, extra-intestinal amoebiasis. The most common site is liver, that is the posterior superior portion, okay, and there is a formation of pus, Encovy sauce pus, and that happens due to the damage to the hepatocytes, and the drug of choice in these particular things for the intestinal amoebasis all is again metronidazole and all. And that depends, pneumonin and extraluminal, dialloxanide furet and all is also being used. Yeah, swimming, very right. Selective IgA deficiency, falling leaf-like motility, brain eating recently Kerala case, okay, of Nagliera fowleri. Tell me regarding this particular fly, which fly it is. As the name suggests, it's a fly. So we are talking about sandfly. This is Phlebotomus urgentipus. It is Phlebotomus urgentipus. That is the sandfly, which is there for the Kala Azar, that you already know. Okay, and important regarding Kala Azar, what I want to tell you regarding that is, in that particular one, what you need to know is that kinetoplast and you go for splenic biopsy and bone marrow biopsy to find the A mastigote form, but the infective form is pro-mastigote. Okay, and important regarding that is RK39 antigen card test. Ranbir Kapoor have turned how much? 39 years. Remember like that. And developed Kala Azar. Yeah, sandfly. Redo-Wit bug. Redo-Wit bug is for the transmitter for the Chagas disease. Aedes and lice you already know. Okay, so Aedes is a number of other vectors like YDRC. Aedes is for what, guys? YDRC. And what is YDRC? Yellow fever, dengue, Rift Valley fever, chikungunya. Okay, and Redo-Wit bug, that you know. Kissing bug, that is Chagas disease. And now we have talked about that. Again, it have come. This is Redo-Wit bug. Okay, this is Redo-Wit bug. Trypanosoma cruzi. It transmits of, does which disease? Chagas disease. Okay, and it leads to that Romana sign, Chagoma, those particular stuff. Megacolon, megaesophagus, myocarditis, meningoencephalitis. Okay, and drug of choice is also important there. That is benznidazole. Aedes, Zika also. Very right. Zika also. Zika is also transmitted by sexual transmission. Right. Now, another favorite question of examiner which is being asked again and again. This one, and they give up these types of history of the oocyst, and they can give this particular size, that is 5 micron. So it comes in the range of 4 to 6 micron, and we are talking about what, guys? Cryptosporidium parvum, which is the most common cause of what? Most common cause of diarrhea in the HIV positive. This is the most common cause of diarrhea in the HIV positive. What? Cryptosporidium parvum, and having the oocyst size is what? 4 to 6 micron. So that also needs to be remembered. Okay, ji? Now, Cryptosporidium parvum is having 4 sporozoites. That also is important stuff which can be asked. 4 sporozoites are there in that oocyst, and the oocyst size is asked 4 to 6 micron. What is the drug of choice for this? Nitazoxanide. And how does it cause a problem? It causes a problem in HIV positive. Okay, it causes a problem in HIV positive. Cyclospora, Isospora belli. These are also there. Cyclospora is having 8 to 10 or 8 to 12 microns. Isospora belli is having oocyst size of 23 to 36 micron, and one more fellow is there which is in HIV positive cause diarrhea, Microsporidium bainusi. So don't forget that particular one. And for these two, cyclo and iso, what is the drug of choice? Cotrimoxazole. Okay. Which coccidian parasite is seen up in this particular picture? They are sickle shaped. Okay, sickle shaped. And the options are these particular ones: bradyzoids, cyst, tachyzoids, or sporocyst. What is the answer? Answer to this question is what, guys? Tachyzoids. They are sickle shaped one tachyzoids and rapidly dividing forms that you already know. Rapidly dividing forms are there, these tachyzoids. Bradyzoids are slowly dividing forms, and they are found within the tissue cyst. Bradyzoids. So if somebody talks about bradyzoids, tachyzoids and all that, you should immediately think for which particular protozoa? Toxoplasma gondii. Okay, Toxoplasma gondii. What is the definitive host here? Definitive host is cat, intermediate host is rat, and accidental host is men. This also needs to know. Okay, and if somebody talks about them, immediately you should have in your mind regarding the toxoplasmosis. Okay, bradyzoid is a resting form, cyst form, and tachyzoids are found in the blood. They are rapidly dividing forms. They look like this, sickle shaped, and like you have those sporulated oocysts in the soil which are infective. Oocyst as such is not infective. So these are infective forms, bradyzoids, tachyzoids, oocyst, but which oocyst? Which is sporulated, that is infective. Okay. Now, if you are observing up these types of things in a surgery, we are talking about hydrated cyst, which is having daughter cyst, granddaughter cyst and all that, and causative organism is Echinococcus granulosus, also termed as dog tapeworm. And what is the definitive host, guys? Definitive host is dog. Intermediate host is sheep. Accidental host is men. And how does the infection happens? By the eggs. Okay, by the eggs. So answer to this question is Echinococcus granulosus, and which D for D, that is dog tapeworm, definitive host is dog. Now, if this particular question comes up in which they show up these striations and this particular thing is there lying, we are talking about Cysticercus of the Taenia solium. And what is the full name of it? Cysticercus cellulose. This is a larva of the Taenia solium. Okay, which is termed as arm tapeworm. This is termed as arm tapeworm, which looks like a Philip shaver. That Philip shaver is Taenia saginata. It looks like exactly a Philip shaver, which does not have hooks and rostrum. Okay, which does not have hooks and rostrum. So we are talking about this. Answer is D in this particular one. And this Taenia solium is only one which leads to this NCC, neurocysticercosis, and it is eating up the eggs rather than eating up the uncooked pork, which leads to more cases of neurocysticercosis. And most common site in that is subarachnoid, and investigation choice you already know, CT or MRI, and drug of choice for that is albendazole. And before that you need to give the what? Steroids. Now this particular egg they can ask you, and this particular egg is having thickenings and filaments in between their hooks. We are talking about H. nana. Okay, we are talking about what? H. nana, which is also termed as dwarf tapeworm, which is also termed as what, guys? Dwarf tapeworm. And this undergoes what? This undergoes autoinfection. Remember this, this undergoes autoinfection, and this is the smallest tapeworm. This is the smallest tapeworm. Okay. Bile stain or non-bile stain egg? It is having a non-bile stain egg. Neha, that mnemonic Neha. What is the drug of choice for H. nana? Answer is praziquantel. The drug of choice is praziquantel, guys. Drug of choice is praziquantel. All these are frequently asked questions. They are being asked again and again. Now this was last year question itself. Having mucus plugs on this, and this is termed as which particular egg? This is Trichuris trichiura, whipworm, having mucus plugs, barrel shaped, dumbbell shaped, and this is the Trichuris trichiura which associates with rectal prolapse in children. This particular one associates with what? Rectal prolapse in children, and this leads to coconut cake-like rectum. This leads to what? Coconut cake-like rectum. Fine. Now this one, having a planoconvex with tadpole-like larvae. We are talking about Enterobius vermicularis, that is pinworm. Pinworm, Enterobius vermicularis, this particular one, and this is the one in which NIH swabbing is done, National Institute Health Swab. Along with that, important point is Enterobius vermicularis, you need to know that it undergoes autoinfection. Like H. nana, this also undergoes what? Autoinfection. Fine. Now, this one with the blastomeres, 1, 2, 3, 4 to 5 blastomeres with like this, they are the features of hookworm egg. They are the features of hookworm egg, and this hookworm egg is non-bile stained. Okay, non-bile stained. And if you just revise up, Trichuris trichiura egg was having a mucus plugs, barrel shaped. Enterobius vermicularis, planoconvex, having a tadpole-like larvae inside. Ascaris eggs still have not come. Then it will come, I will tell you how does it looks like. So this is hookworm egg, fine. Also termed as Ankylostoma duodenale egg. Now this is the Ascaris egg. This particular one is Ascaris. This is the fertile egg, which is having a proper shape. Okay, thick albuminous coat is there, whereas this particular one is having elongated shape. This is having elongated shape, and this is termed as infertile egg, and this is decorticated egg. Fine. So Ascaris egg. How does the infection of Ascaris happens? By eating up. We have a mnemonic na for eat. By these eat. Enterobacter vermicularis by eating the eggs, Ascaris by eating the eggs, and the Trichuris trichura by eating the eggs. So these are there in which the infection happens by eating the eggs. Is it fine, guys? I know a bit fast I have to be if I want to cover up important aspects. So excuse me for that. But we need to be slight bit fast. This is another favorite one. Banana shaped gametocyte. That is a feature of which particular one? Plasmodium falciparum. And it's a macro gametocyte. It can be a micro also. But the options is written macro, so okay, macro. Banana shaped gametocyte is a feature of it. And sporozoites, that is this particular one. And regarding this particular stuff, Plasmodium, which is the most dangerous, Plasmodium falciparum. Okay, and definitely first here is female Anopheles mosquito because sexual cycle happens in that. Fine. And regarding the Plasmodium falciparum, already I have told up a mnemonic umber. Okay, and in this umber, what all you have? A is all age RBCs are involved up. Okay, Morris dots are seen, banana shaped gametocytes are observed up, Ecol forms are seen, recrudescence is there, and the renal failure is there. Okay, and the renal failure is there in this particular one. All these are important features of Plasmodium falciparum. Okay. Now, this is Wuchereria bancrofti, and this is the microfilaria of it. And as you can see that in the tail tip, there is no nucleus in the Wuchereria bancrofti. And how to remember that? Remember like this, there is a ban on the nucleus. And terminal 2 nuclei is a feature of Brugia malai. Okay, Brugia malai microfilaria is having what? Terminal 2 nuclei. That also needs to be remembered. Now, which form of hookworm is this? Which is having a needle-like stuff. And needle-like stuff is a feature of filariform larvae. Okay, filariform larvae is needle-like, and that's why this enters the sole of the foot. In the case of Ankylostoma duodenale, okay, in the case of strongyloids, and in the case of Necator americanus, that is ANS. In all of them, the portal of entry is sole of the foot, like that, ANS. Okay, so this is filariform larvae. This one, this have entered up. See this. This has entered up into the sole of the foot and could not able to penetrate all the skin layers. That's why you are seeing up here. This is cutaneous larvae migrans (CLM) done by Ankylostoma brasiliensis. And if it is having a history of dog, then it is caninum. And even strongyloids stercoralis, this also leads to larvae currents, which is also a type of what? Cutaneous larvae migrans. And the drug of choice for this is L-bendazole. The drug of choice for this is L-bendazole. Okay? Now this skin lesion, as it is seen, depressions are there. We are talking about umbilicated nodule, and that is observed in Molluscum contagiosum, guys. Molluscum contagiosum, this is a pox virus. This is a pox virus, and mostly they are done by MCV1. MCV2 causes infections in HIV positive ones. So pearly white umbilicated nodule is a typical feature observed in Molluscum contagiosum disease, and important is molluscum bodies, also termed as Henderson-Peterson bodies, which are intracytoplasmic inclusion bodies. They are typical of it. That is the Molluscum contagiosum stuff. Okay. Now, anything which is aggregating like this, multinucleated one, they are becoming up. We are talking about the Zhang smear. And Zhang smear is done for HSV1, 2, and remember it is also done for HHV3. It is also done for what? HHV3. And what is HHV3, guys? Varicella zoster. That is the chickenpox one. Okay. Now, this everybody knows that the virus responsible is Varicella zoster, that is HHV3, and this is herpes zoster ophthalmicus. This is the reactivation of this herpes zoster ophthalmicus, this one. And single-sided lesion is observed in that, also termed as herpes zoster or shingles, right? And this is reactivation of this particular sort only. Now, this cute child is being slapped. So slap cheek appearance, that is being done by which virus? Parvo B19. This was INICT favorite this time. It leads to P for P, pure red cell aplasia. It leads to what? Pure red cell aplasia. Fine. This particular one associates with pure red cell aplasia. Other than that, it associates with aplastic crisis in sickle cell anemic patients. Okay. It associates with NIHF, non-immune hydros fetalis. Okay. And polyarthralgia syndrome. Polyarthralgia syndrome. And the question depicts of what, guys? Erythema infectiosum. Okay, erythema infectiosum, also termed as fifth disease, having a slapped cheek appearance. Okay, having a slapped cheek appearance. And how to remember that? Remember like this: apne kya ho gaye? Paraye, after a slap. Okay, and what is this A? A is aplastic crisis, P polyarthralgia and pure red cell aplasia, N is NIHF, non-immune hydros fetalis, E is erythema infectiosum. Okay, so these are the important ones which you should know before going in the exam. Now, this particular one is what? Which virus does these genital warts? Obviously, you know it is HPV. And which particular ones? Mainly 6 and 11. HPV 6 and 11 leads to what? Genital warts. Parvo 19, just now we have done. Pox virus, molluscum contagiosum 1, herpes, we have already discussed now. Okay. So, HPV 6 and 11 is this. And 6, 11, 16, 18, they can lead to CIN and carcinoma cervix also, that you know. And in this, important is this one, E6 and E7. And they cause the loss of which particular gene? It does the loss of P53, and this leads to the loss of RB gene. And these are tumor suppressor gene, their loss leads to tumors. Okay, how to remember? 6, you tilt it, it looks like a P, 7 colors of rainbow, that is retinoblastoma stuff. Okay, so that also needs to know. Okay, the answer is HPV here, human papillomavirus, and it is a DNA virus. It's a DNA virus. This particular Purkinje cells, Negri bodies, if this word comes, then there should be no doubt regarding the rabies virus. Okay, what is the speed of rabies virus? Condylom equinatum, you are right, delirium. Okay, rabies virus speed 3 mm per hour. This is the speed of it. Fine. Which particular two places that does not have rabies in India? Andaman, Nicobar and Lakshadweep. Water isolated areas, they do not have it. And that you already know, that furious rabies, dump rabies and all that stuff. Important is from where you take the sample for the antenatal diagnosis. For the nape of the neck hair follicle, this is better one, and corneal smear, and you go for those fluorescent testing here, and you can go for the PCR also. And in the post-mortem ones, you have to go for this brain biopsy from the hippocampal area. In that, you observe these Negri bodies, which are intracytoplasmic eosinophilic inclusion bodies. Fine. Now, Nipah, if you are seeing a bat, in the picture, along with the pigs, along with the men standing there, immediately what should come in your mind? Nipah virus. And which state should come in your mind? Kerala. Okay. So, Nipah was there. Okay. So, this is what is Nipah virus, and if these pictures are observed in your exam, immediately you should think for what? Nipah. Can you tell me, Nipah is a member of which particular family? If you tell this, you are about 200 for sure. Let me tell you. If you tell this, it's a, yes, tell in the chat box. It's a Nipah virus, it's a which family member? Paramyxoviridae. It is which one? It is Paramyxoviridae. Fine. It's a Paramyxoviridae having mortality of more than 70%. Now this, everybody knows, this is Aedes tiger mosquito, and that leads to YDRC we have just discussed up, along with Zika, which you have told up. Fine. This is a light microscope, and as you can see, they have asked once, where is the condenser? Down is the condenser. Okay, this is eyepiece through which you see, and this is objective lenses. It can be 10x, it can be 40x, it can be 100x. 100x is oil immersion ones. Fine, and these are all common sense things. These are objective lens, this is eyepiece, and this is condenser. Condenser they have asked up once. Now, anything which is spherical, which is filamentous and like that, we are talking about hepatitis B virus. Okay, it exists in three forms: spherical, tubular, and Dane, which is the most common, spherical. Okay, what is the serological marker in the ELISA for the acute hepatitis B? So guys, the best answer is HBsAg positivity along with IgM NT-HBC. This is there. Okay, this is acute hepatitis B. More than 6 months, if somebody is HBsAg positive, that is termed as chronic hepatitis B. So that also needs to be remembered. Okay, and if you are getting only the antibody titers against the surface, we are telling that that fellow is vaccinated. And that should be more than how much? More than 10 milli international units per ml. Okay, that should be there, and that is termed as vaccinated. And the vaccine is prepared where? In the East. And 016 is the schedule for it. 016 is the schedule for it, and hepatitis B virus is the only virus which is a DNA virus here. Otherwise, most of them they are RNA here. Okay, and this time they have asked up regarding the incubation period in the INI-CET exam. So this can come to you also. Very simple trick is there. Hepatitis A is having the incubation period of 15 to 50 days. Hepatitis B is having 50 to 150 days. Okay? Hepatitis C is having, this 50-50 cancels, 15 to 150 days. Hepatitis D is same as B, and hepatitis E is same as A. Simple. Okay, 15 to 50 days, 50 to 150 days, 15 to 150 days, and this particular one, hepatitis E, is 15 to 50 days. Okay, so remember this particular one. Like this, you can remember of this. But this 50 is actually what? 45, and this 150 actually is what? 180. Okay, but you could able to recall the things by this 50. 15 to 50, 50 to 150, 15 to 150, that 50 is actually 45, and that 150 is actually 180. And if you remember like that, your things are sorted. Okay. Now, this was asked in the this Fmge exam itself, and obviously when you're seeing up GB120, GB41, all these integrase enzymes and all reverse transcriptase, we are talking about which particular virus? Obviously it is HIV. Single stranded, double stranded, single stranded. Okay, this is a single stranded RNA DNA RNA virus having envelope. This is an enveloped virus. Okay, this is a enveloped virus. Anil, तुम इस बार पास हो, 200 के around हो. Fine. Now this is Zika, and you can see that this particular child is having microcephaly, and the pregnant female, they're having those Hofbauer cells in the placenta which are affected by this Zika, and this is transmitted by Aedes, as you are telling up. Sexual transmission is there of this, and microcephaly. And another important association is GBS, Guillain-Barré syndrome, which strongly associates with which particular bacteria also? That is Campylobacter jejuni. And another one, Mycoplasma pneumoniae. Okay, Mycoplasma pneumoniae also. This is which virus? This is Ebola virus. This looks like a Ebola virus. Ebola virus is the one which needs biosafety level 4. This is dangerous. Dangerous. Okay, more than 50% mortality is there in this Ebola. Zaire Ebola is the most virulent. Ivory Coast is the least virulent. Also transmitted with sexual transmission is seen in this also. Now, this is that famous one which have changed the life of all of us, and that is COVID-19 virus. Which one? SARS-CoV-2. Single stranded, double stranded. It's a single stranded one. RNA, DNA, RNA. Okay. Segmented, non-segmented, non-segmented. Enveloped, non-enveloped, enveloped. Negative sense, positive sense, positive sense. Which particular important virus is negative sense? Influenza virus. Influenza virus is negative sense. Remember that. Okay, and that is segmented also. That is having influenza A, 8 pieces of RNA. That is also important. This is corona. This is SARS-CoV-2. Doing up this coronavirus disease 2019. This particular variant, Omicron variant, what is the number of it? B.1.1.1529. This is the Omicron variant. 1617.2. If somebody writes up like this, 1617.2. This is the Delta variant. Might be asked. What is the investigation of choice? RT-PCR. Okay. RT-PCR. In which particular biosafety levels you do it? That is biosafety level 2. Okay. And what about the culture of it, by which you are able to make the vaccine of it? The vaccines are being prepared up in the BSL-3, C for C culture, hai na? Third alphabet. Then which particular vaccine of ours is using up adenovirus, chimpanzee adenovirus, Covishield, which is having the inactivated virion, that is the Covaxin. Okay, which is the worst vaccine in the world, that is the Chinese one. Sinovac, Sinopharm, they are the worst. mRNA vaccines, Pfizer, they are said to be the best. But God knows what is best. Okay. So as this is seen up, that spike proteins and all that is there. So you already know that all these are there in this. Now this everybody knows. If you are seeing up a bullet shaped stuff, we are talking about which virus? We are talking about the rabies virus. We are talking about what? The rabies virus, and the shape shape is that this particular one, and this is the one which is also negative sense. This is also a negative sense form. What is the meaning of negative sense? Negative sense means that firstly it will change to positive sense and then it will replicate. So this rabies virus is also a negative sense virus. Nasal swabbing, that you know na. Nasopharyngeal swab and oropharyngeal swab you take. Good. What is this, guys? This picture is regarding the autoclave. Okay, an autoclave is which sterilization? It is moist heat sterilization, and more than how much? More than 100? 121 for 15 minutes at a pressure of 15 psi. This is regarding the autoclave stuff. 121, 15 minutes, 15 psi. Moist heat sterilization. What is the control used in it? Control is Bacillus stereothermophilus. Okay, that you need to know. Geobacillus stereothermophilus is also another name of it. That is the control used. Frequently asked question, that is. Besides that, what do you autoclave? Sputum samples. Before discard, you have to autoclave. Surgical instruments, certain, you have to autoclave. Okay. Besides that, all the culture medias, all the surgical sutures, except the catgut, they all need to be autoclaved. Fine. Below 100 is inspeciation, pasteurization. At 100 is tyndalization and boiling. Above 100 is autoclaving, and this is moist heat sterilization. Moist heat is better than the dry heat. Dry heat, typical example is hot air oven, 160 for 2 hours, and the control used is what there? Bacillus subtilis and atrophius. Nowadays, if this particular option you get, Bacillus atrophius, so that should be the answer there in that particular one. Fine. And then you have those incineration, and which particular bags are incinerated, guys? Yellow colored bags are incinerated, and flaming is also one of the dry heat sterilization. Those things which you can't heat, you have to filter them up. Okay, ionizing radiation is which one? The gamma rays and all. Non-ionizing is which one? Those UV rays and infrared rays. Okay, what do you use in blood spills and all? 1% sodium hypochlorite. Fine, 1% sodium hypochlorite. That ethylene oxide gas, which is very highly effective sterilant. It is a high level disinfectant that also needs to be remembered for catgut sutures now, and for the those pre-packed syringes. If somebody asks now which is better, so that thing is better for that. Fine. Pre-packed syringes. Besides that, those names, Riedel-Walker coefficient test, Kelsey-Sykes test, okay, Chick-Martin test, in-use test, they are the test for the efficacy of disinfectants. That also is to be remembered properly. Fine. This is erythroblastosis fetalis, as it is written of. Which hypersensitivity, guys? This is type 2 hypersensitivity. RH incompatibility, erythroblastosis fetalis, along with the autoimmune hemolytic anemias, transfusion stuffs, they all are coming in which particular one? type 2 hypersensitivity. Okay, along with that, myasthenia gravis and Graves' disease also comes up in the type 2. They have another type, but still they are under what? Type 2 only. That is cytotoxic one. Anaphylactic, you already know that any sort of allergy, that is termed as anaphylactic one. Which interleukin is involved in the type 1? Interleukin 4. Which helper cells are involved up? T helper 2. What is the mnemonic? 1, 2 ka 4. Means type 1 hypersensitivity, interleukin 2 and interleukin 4, and which helper cells? T helper 2. 1, 2 ka 4. That is T helper 2 cells, they secrete up interleukin 4, which attract which particular immunoglobulin? IgE in the case of type 1 hypersensitivity. Cytotoxic one, the antigen is a part of the cell, to that antibody attaches up, there is a complement activation and complement does the damage. Fine. And in the type 3, that is immune complex ones, the antigen is not bound to any cell surface, it is free and it attaches with the antibody, produces immune complexes, activate complement, and most of the autoimmune diseases, they are there in the type 3. And typical examples are Arthur's reaction and serum sickness. Along with SLE, PAN, all that is there in the type 3. And type 4, sensitized T lymphocytes, that is involved up. They secrete up lymphokines, and there is a formation of granuloma in that. There is a formation of what? Granuloma, tuberculin test, lepromin test, contact dermatitis, a different type of type 4 hypersensitivity. Otherwise, mostly allergies, they are there where? In the type 1. I suppose this particular thing helps. And interleukin 2 plays a role where? Interleukin 2 plays a role in type 4. Okay, and which helper cells? T helper 1 secretes interleukin 2. That is important. Okay. So 1, 2, 4 is for type 1 hypersensitivity. So guys, this much is there. If you revise up this much only, it will touch upon 30 to 40 for sure. And you can very well see up in the exam also that if you have a sound knowledge of this, then you can apply this at a several fronts and all. Okay. And in the last boosting for you, I like to tell this only that it's all the game of mindset. Okay. So it's all here. Nothing is impossible. Impossible itself is telling what? I am possible. So you have to just analyze yourself that where I am going wrong, what should I do in the last days. Okay, if I am having any anxiety issues and all, how to tackle them up, how to go for a wonderful exam on that particular day. Okay, so prepare your strategy for that particular day, that how to go for that and in which way you will go for the exam, and which particular thing firstly you will answer up, and you will not panic if you don't know the answer and all, because that doesn't matter. The question is exam is of two sessions. So even though one session goes down also, you have to score up 150 itself. You can easily score up if one particular aspect is wonderful. Okay, but yes, good going is always good. And I know my personal experience is there that I have seen up those students who are passing up in the 8th attempt also, and only zeal is that they have not left it. Okay, हारते कब है हम? जब हम छोड़ देते हैं. तो छोड़ना नहीं है, लगे रहो, ठीक है, और अपना बेस्ट दो. And very best of luck from my side. Thank you so much. I hope this particular session is useful for you. So thank you so much from my side, and do revise it up again. Bye.